Hey, you guys. Sorry this lecture is a day late going up. I will always, of course, endeavor to have the lecture up before or on the day that's listed in the schedule, but I've had to implement a new homeschooling program for a sixth grader, and we don't always get started on time. So thank you for your patience, and let's continue on with our third vision lecture and talk about color vision and the cone options. So some of you might recognize this little guy over here. This is a mantis shrimp. And as you probably know, and we'll revisit today, we have three cone options. And that gives us a pretty spectacular range of colors that we can visualize. The mantis shrimp expresses not 4, 5, 8, 10, but 16 cone options. So they're under investigation for a number of reasons. Their massive strength relative to their size is number one. So they're ferocious predators. And number two, their vision, we simply can't imagine what it would be like to have 16 cones. It would be like trying to imagine a million colors you've never seen and then try and imagine a million more. Okay. So interesting little creatures, and we'll talk about cones and color vision today, building on the receptive fields. So if receptive fields are still fuzzy, go back because we'll be building on receptive fields in the context of cones and color vision today. And again, I'll guide you or I'll direct you to the TUDIS supplementary files. There's something useful in there for all of the sensory systems we've discussed, but it's particularly good for vision and receptive fields and color vision. So go there. There's a folder in our supplementary folder called the TUDIS supplementary files. So that's a good alternate source of information if you need something outside of the lectures for this set of information. All right, so we'll talk about the three human cones, their structure, and their spectral sensitivity, meaning the wavelength or wavelengths to which they're most sensitive. And we'll talk about three different aspects of color vision: contrast, constancy, and assimilation. And that'll be easier when we look at some specific examples. Center surround receptive field, you should already be able to do that. Diagram a basic center surround receptive field, naming the key cellular components. That should be review or study from last lecture. But we'll build on that to look at center surround receptive fields for color vision. And that'll bring us to cone opponent ganglion cells. There are opposites. There are color opposites. Red and green are color opposites, and blue and yellow are color opposites. Okay? Green and red are color opposites. Blue and yellow are color opposites. And then we'll talk about pathophysiology. So color blindness or red-green color blindness is fairly common. So we'll talk about that and a couple of other conditions that are associated with missing or broken cone options. All right, so this should be review. We started with the molecular physiology of the retina, and we built on this when we talked about receptive fields last time. But let's just remind ourselves of the important cells that are here. Right? And the path of light, remember, is in this direction. The path of light is going from the pupil towards the back of the retinal pigment epithelium, and signaling is going in the opposite direction. The first cells that are activated here are the photoreceptors, and those signals are passed to the bipolar cells and ultimately the ganglion cells. We know that horizontal cells and amacrine cells are also here. Horizontal cells are important for lateral inhibition, I hope, is the answer that you would give me. So light enters the eye, is focused on the retina, specifically at the fovea of the retina. The focal point is the fovea. Within the fovea, there's a high number of cones, and that should make sense because the fovea, the focal point, that's responsible for high acuity, sharp color vision. That's where the cones are. So there's a relatively low number of rods, virtually none, relative to the rest of the retina. So the extrafoveal regions, the periphery of the retina, if you will, contains few cones, and many, many rods. And remember, the function of most of the retina is giving information on intensity of light, ambient light, how much light. Whereas at the fovea, that's sharp, high acuity color or scotopic vision, right? Rods are responsible for photopic vision, yes or no, how much light, and cones are responsible for scotopic, color, high acuity vision. Okay, and then just some nice fluorescence imagery here. You can see the rods and the cones colored in green, the various different types of bipolar cells and amacrine and horizontal cells, and then ultimately the axons of the ganglion cells converging into the optic tract at the optic disc. We haven't looked at a lot of immunostaining and fluorescence in this particular course, but it's certainly a nice image where we can, instead of a cartoon, visualize what these cells would actually look like. And I believe this was mouse retina. Okay, so the cone opsins, like rhodopsin or rhodopsin, are all GPCRs. Rhodopsin remains the best studied, but we know a lot about cone opsins as well, and they're quite similar, right? Qualitatively, all of the signal transduction pathway for rods and cones is similar. There might be some quantitative differences in kinetics and certainly wavelength sensitivity, but largely the signal transduction mechanisms that you learned for rhodopsin also apply to the cone opsins. G protein is still transducin. They all contain a retinal binding site by bovine rhodopsin numbering, that's K296, but of course the actual location of the residue, maybe it's K292, maybe it's K298, but somewhere around K296 there is a critical lysine residue, and remember that's important because that's where the retinol chromophore, shown over here, hiding in the background, binds. And then what about the differences in spectral sensitivity, right? We know that rhodopsin is maximally sensitive at about 500 nanometers, but the blue and green and red cones express opsins that have peak wavelength sensitivities in those color regions. So what accounts for that shift? And what accounts for that shift is the binding pocket around the retinol will differ based on changes in amino acid sequence. So there's a lot of homology between rhodopsin and the short, medium, and long wavelength sensitive cone opsins, but there are some differences, and we don't care about the exact amino acid sequence differences. However, hopefully you can appreciate that based on the functional groups, the R groups that are present on the amino acids that are facing into this binding pocket, the properties are going to change. And if the properties of the binding pocket change, then the properties of photoisomerization of the 11-cis retinol are also going to change. So the wavelength sensitivity will be a function of the specific amino acid residues and dependent on non-homology or sequence changes between the various cone opsins. So you would correctly refer to long wavelength sensitive, medium wavelength sensitive, or short wavelength sensitive opsins. So I don't want you to refer to green and red and blue opsins. You can refer to green and red and blue cones, right? But you would correctly say that a blue cone expresses a short wavelength sensitive opsin. A green cone expresses medium wavelength sensitive opsin. Red cones express long wavelength sensitive opsin. And rods express rhodopsin. So just make sure you're clear on that terminology. Okay, so we saw this slide last time. Let's just remind ourselves along the electromagnetic spectrum, we're increasing in wavelength over here from UV, which we can't see, blue, green, yellow, and red, and then if we go further, we'd be up here into infrared, which we also can't see because we don't have opsins that are sensitive or receptors that are sensitive to those wavelengths. Can we see them? Yes, but we'd have to use the appropriate filters to do so. So you should know these approximate numbers. Short wavelength sensitive, so I've denoted that with an S up here, expressed by blue cones, maximally sensitive around 420, medium wavelength sensitive around 533, and long wavelength sensitive around 560. And hopefully I remembered to mention last time that 564 is actually in the yellow part of the electromagnetic spectrum, and red would be over here. However, the red cone expressing the long wavelength sensitive opsin is the one that will be most sensitive to red, and the one that will be really appreciably sensitive to red. So that's why it's referred to as a red cone and not a yellow cone. Okay? Because you can see for yellow, there's overlapping. You'll have activity, your medium wavelength sensitive opsins and your long wavelength sensitive opsins will both be giving information on within the yellow region. So that's why it's referred to as a red cone and not a yellow cone. And that reminds me of another important point, which is there's a range of wavelengths over which these opsins are differentially sensitive. So is it that nobody's giving you information at 475? No, at 475, you're certainly getting information. It's largely from blue cones, but green cones might be giving some information too if we're at 475. So it's important to realize this is a spectrum, right? You're looking at the electromagnetic spectrum, yes, but there's also a spectrum of cone activity at any given wavelength along this spectrum. The relative activity of the relative excitation of the various opsins is what allows us to see different shades, hues, and so on. So activating the opsins in different combinations to different degrees at different wavelengths allows us to have so-called trichromatic vision, right? You can see a large number of colors and hues with very few, just 3 cone opsins. And remember, the rods aren't contributing to color vision because rods only express one opsin, and that's maximally sensitive at 500, and that, of course, is rhodopsin. All right, so trichromatic vision, another shrimp mantis down here, and the rainbow of colors that we can see. And so just a reminder that if we can see this substantial rainbow of colors, imagine what a mantis shrimp might see with 16 cones if we only have 3. And so it's not quite the same as mixing paint, but it's kind of similar to mixing paint, right? Different opsins are expressed in different combinations to different degrees, and that creates a palette, almost like an artist mixing paint. However, when you mixed paint as a kid, or maybe you still mix paint, in order to make green, you would have mixed blue and yellow, right? Blue and yellow are primary colors when it comes to paint. However, in the visual system, you get yellow by mixing red and green. Okay, so when you activate red cones and green cones at the same time, that gives the perception of yellow. Okay, so similar to, but not quite the same as mixing paint. And we have really 4 primary colors when it comes to vision: red, blue, green, and yellow. And remember cone opponency. Red and green are cone opponent or color opposite, and blue and yellow are cone opponent or color opposite. And what I mean by that is if you close your eyes and you think of red and green, and now try and think of a color that is reddish green, right? There is no reddish green. You could think of a reddish yellow color, right? And a reddish yellow color might lead you to orange, but there's no reddish green. Likewise, blue and yellow are cone opponent. They're color opposite. If you were to mix, if you were to try and think of a bluish yellow color, well, you can't really do so because there is no bluish yellow. Okay, let's continue with contrast, constancy, and assimilation. So contrast is what we were discussing on the previous slide. It's the difference in the wavelength or the luminescence of an object in your field of view compared to other objects within that same field of view. and we'll come back to cone-opponent receptive fields, but hopefully I've alluded to this already. Contrast specifically between color opposites. Yellow is always going to stand out against blue, and red is always going to stand out against green, right? If somebody were standing far away from you and they were wearing a blue shirt with a yellow tie, you would always be able to see that striking yellow tie against the blue shirt. However, if somebody was wearing, let's say, a purple tie on a blue shirt, at some distance you would be able to distinguish blue from purple. But as that person got further and further away, those two colors might blend together. Okay? But blue and yellow are always going to stand out, and green and red are always going to stand out as color opposites. Okay. So this is a good example of contrast. Constancy and assimilation, I'll just define them for you now, and then we'll come back to some examples after we spend a little bit more time talking about contrast and cone opponency. So constancy is our ability to determine the color of an object irrespective of the amount of luminescence. So if I was wearing a red sweater in the lecture hall in full light, you would be able to tell that my sweater was red. But if we were in a dark movie theater, even though the ambient light was low, you're still pretty good at determining that my sweater is red, okay, regardless of the level of illumination. Of course, if we're in pitch black, you would lose that ability, but throughout a fairly large window of luminescence, our constancy, our ability to perceive a color and predict what that color is fairly constant. Sometimes it fails, and if you remember back earlier in the course, I showed you a picture of the dress, and we'll come back to the dress, but the dress is an example where constancy fails. And then assimilation is a tendency for a color to take on hues of colors that are surrounding it. And that'll become clearer again when I show you an example towards the end of lecture. Okay, another painting by one of my favorite artists, Georges Seurat. This one is in color. He still uses pointillism, but he achieves the same effect as he does with black and white here by juxtaposing the yellows and blues, right? So without knowing the circuitry of the retina and without knowing about cone opponency, Seurat plays into accentuating colors that are cone opposites, red and green and blue and yellow. Okay, I saw this at the Musée d'Orsay in Paris last summer, which feels like another lifetime at present. In any case, I hope you too have the opportunity to visit Musée d'Orsay in Paris at some point. All right, let's come back to receptive fields. And this, I think, is a very friendly diagram that hopefully looks familiar. We spent a lot of time on this. We used slightly different depiction, but all of the key parts are here, right? Let's just review. We have a center and we have a surround. At the center, we have a cone and we have a bipolar cell. In the surround, we have cones and we have horizontal cells. Okay, so the center is made up of cones and bipolars. The surround is made up of cones and horizontal cells. And you should be able to walk me through an on-center, off-surround, and you should be able to walk me through an off-center, on-surround. And I'm not going to review all this circuitry again because I'll be repeating myself. And if you need to, you can go back and re-listen to the previous lecture. But whether it's an on-center or an off-center system will depend on whether or not there is an on-bipolar cell or an off-bipolar cell at the center. So very quickly, if we have an on-bipolar cell at the center, you'll recall that on-bipolars express M-gluars, which are inhibitory, and TRPM1 channels, which are depolarizing, but only when they're open. And we shine light on the center. We're going to hyperpolarize our cone. That'll decrease glutamate release. That will reduce M-gluar activity. That will open up TRPM1 channels. That will depolarize our bipolar, increase glutamate release onto our ganglion cell, and increase firing. in our ganglion cell. Let's do the same thing and shine light this time on the surround. When we shine light on the surround of an on-center, off-surround system, we will hyperpolarize our cone. That'll reduce glutamate release onto our horizontal cell. Remember, this releases GABA, so that'll decrease GABA release onto the center cone. Center cone is in the dark, will do what cones do in the dark and increase release of glutamate onto our on-bipolar cells, increase mGlu-R activity, inhibit TRPM1, hyperpolarize, and we get less firing in our ganglion cell. I will leave you to do the same exercise for an off-center on-surround using this type of circuitry. Go through and do what we just did, shining light on the center and shining light on the surround of an off-center, this guy over here, of an off-center on-surround. Now I want to build up just a little bit onto cone opponent retinal ganglion cells. So we could think of 4 different types of cone opponent retinal ganglion cells. And remember, we're talking about the receptive field of the retinal ganglion cell, which will be made up of the bipolar cells, the horizontal cells, and the various cones in the center and in the surround. So you can imagine a green on, red off. The opposite would be a red on center, green off surround. A yellow on center, blue off surround. And the opposite would be a blue on center, yellow off surround. And I don't need you to memorize the circuitry for all 4 of these. I want you to get the big picture, which is we've been talking generally about having cones in our center and our surround. But what about now if we have specific types of cones in our center and our surround? Would it not make sense that we could make this center sensitive to green, right? And we could make this surround sensitive to red? Well, we could, depending on the type of cones that make up our center and our surround. So my suggestion will be choose your favorite and be able to describe one of these in terms of the circuitry. And keep in mind, the yellow might be a little more complex because what do you need, right? If we were in lecture, I would ask you, what do you need to get yellow in the visual system? And you would remind me that you would need to have both red cones and green cones. So these 2 will be slightly more complex in terms of the circuitry because you'd need 2 different types of cones present in the center or the surround in order to get yellow. So I like this one. Let's go with a green on center, red off surround. 2 key components, as always, the type of bipolar cell that's at the center. If this is an on surround, then same story. If we have an on center, then this is an on center system. Okay, so our bipolar cell matters, still expressing mGlu-R and TRPM1. And then the other key component is the type of cone. So if this is a green on center system, and then I'll ask you to predict the type of cones that are at the center, and you would tell me, hopefully, that those are green cones expressing which opsin? Medium wavelength sensitive opsin. Okay? And then the story is the same. If you shine green light on the center of a green on center system, green light will hyperpolarize this cone. That will decrease glutamate release, decrease mGlu-R activity, increase TRPM1 activity, and increase firing. Let's shine red light on the surround of our green on center, red off surround. Okay. The surround cones are sensitive to red. Right? These would be red cones expressing long wavelength sensitive opsin. We shine red light on red cones, they're going to hyperpolarize, decrease glutamate release onto the horizontal cell, which will decrease glutamate release onto the central cone. Central cone is effectively in the dark, doing what cones do. spitting out glutamate onto the bipolar cell, increasing mGlu-R activity, decreasing TRPM1 activity, decreasing glutamate release onto the ganglion cell, which will decrease firing. So when you shine green light on the center of a green on-center system, you will turn it on. When you shine red light onto the surround of a red off surround system, you will turn it off. You will inhibit firing of a retinal ganglion cell. And it's fine to memorize that statement, which you can do simply by looking at this simple diagram, but I need you to be able to walk me through the circuitry, right? For one cone opponent system, choose your favorite. I've made it easy by giving you this one here. If you want to choose a different one or practice other ones to really nail this home, then by all means, please go ahead and I encourage you to do so. Okay, so what's the point? Let's just stare at the X and talk about why this matters. So right now, if you're looking at the X in your foveal field of view, there are all of the cone opponent fields that we just looked at. And in about 5 seconds, I'm going to switch the slide to present a blank page, and I want you to tell me what you see. Well, you can't tell me what you see, but anyway, focus on where there is, look at the X, but think about where there was yellow, right? Cool. What do you see where there was yellow? Where there was yellow, you see blue. What do you see where there was blue? At the bottom, you see yellow. And it's fading now, but you can rewind me and do it again if you like. But this is what happens when you, well, this demonstrates cone opponency. And so what happened is your cone opponent fields became bleached in that 30 seconds where you were presented with the 4 different colors. And when you're now presented with full spectrum of light, right here, white light, the other cones are more available. Right? So where there was the blue spot, your blue cones or some of your blue cones had bleached out. The ones that were most available to respond to the white light in those receptive fields were the yellow ones. So you perceived yellow even though there was no yellow in the presence of white light. Okay, so it's a good, hopefully good demonstration of cone opponency. All right, I need to take a quick break, but I will be back to do part 2 of this lecture shortly. Enjoy the break and stay safe, and I'll see you soon.